Hi and hello students, welcome back to another session. Previous session we have seen what are the different aspects of blood, that's the components and what do they contain. And in this session we are going to look at the blood vessels, the blood groups and the clotting factors. That will be our topic. In that we are going to look at how blood clots and how the blood vessels carry the blood to various parts of the body and what are the different kinds of blood groups we come across in human population. With that introduction, let's get into the subtopics. So today's topics will be what are blood groups and how exactly we understand how they are formed and why is clotting very important and what is a clot. Now how blood is circulated inside the body. Now these will be our subtopics for today. Now coming to the next one. On the screens, we are having what is clotting of blood, or we simply call it as blood clotting. So, we must have experienced somewhere or the other when we had an injury that there is always blood loss. Now, blood loss cannot happen for a longer period of time because we end up in losing a lot of blood. So, what do we do then? So, we wait for a minute or two and then we see the blood turns dark in color. And then slowly the flow of blood stops. Now what is that called as? That is nothing but the clotting which happens during an injury or during any kind of surgery and all that. You need to clot your blood. Now coming to the topics, what we are looking at here, how clotting occurs. The first thing, what are the factors which help in clotting? So previous session we saw that there were something called as the thromboplatelets or the thrombocytes, which are a part of the cellular components of blood, and this helps in clotting of blood. So we have already seen this before. Now, how these platelets exactly help in clotting of the blood, we will have a look at that procedure. Now coming to the first one. So here on the screens you have a flow chart as such is given here. Now what is the flow chart about? We will try to understand. So whenever there is an injury here, you find the blood platelets are coming to your rescue. So it has to come because that is how the clotting occurs. Now the liver gives out the vitamin K and here you can see there is thromboplastin. Thromboplastin is an enzyme otherwise called as thrombokinase. Now why do we need this enzyme? Now this enzyme along with vitamin K helps in the production of calcium ions, and this calcium ions will activate an inactive form which is called as the prothrombin. Now why prothrombin has to be converted? Because only when it takes the vitamin K from the liver and the calcium, it will be converted to thrombin, which is an active form which initiates the clotting process. So till here, till the thrombin is formed, there is no way that the clot can happen. Now this will be the first part of blood clotting, and clotting can otherwise be called as coagulation. Another name given to clotting will be coagulation. So we can also call it as the first phase of blood coagulation. Now once the thrombin gets active, the fibrinogen, when we did the plasma previous session, we saw that there were certain proteins and clotting factors. Now this fibrinogen is in an inactive form in the plasma. Now this has to convert it to an active form so that actually the clot forms at that site. Now fibrinogen along with thrombin, it converts into a protein called fibrin. Now this fibrin is the one which comes to the rescue and forms a clot. And how does the clot form? The clot forms like a mesh. We call it as a network or a mesh-like structure is formed at the site where clotting takes place. Now that mesh will relieve you from the blood. And you also have the dead RBCs, the dead bacteria and the dead WBCs. Now before I understand, the initial part we said that it should start the initiation of that process. Now that will be called as a platelet plug. Whenever the blood platelets enter, they give out the platelet plug, and this plug is to stop the flow of blood at that particular time. So what happens? The blood cells all start clumping in that particular region. like the one you're seeing on the other side. They all clump into that region so that the blood flow stops then and there. Now that is the purpose of having a platelet plug. Now we can always ask that why platelet plug is different from a clot. It's not like that. Platelet plug will just help you to stop the flow of blood. But the clot which is forming later on, will help you to remove all the unwanted substances and protect you from that microorganisms. Both happen simultaneously and both are different. Okay, so this is all about the clotting of blood, and onto the screen you saw that this is the mesh-like structure we were talking about. Now this is because of the fibrin which is being released at that clotting area. Now this is clotting of blood, otherwise called as coagulation. Sometimes people do not have these clotting factors. Now when they do not have clotting factors, they develop a disease which is called as the hemophilia. Now what happens in hemophilia? Some of the factors of clotting are missing, so they will not have that capacity to clot in the initial period of time. For such patients, you have to incorporate or give the clotting factors from the outside. Now unless you do that, the clotting never stops. Now this is called as the bleeder's disease. So hemophilia otherwise can be called as the bleeder's disease because it keeps happening and the blood flow keeps moving forward. So that's the name given for hemophilia. This is a disease where clotting factors are absent. Now coming to the next one, what we look on the screens is the blood vessels. So we just know that blood flows throughout our body. But have we ever imagined, so what are the different kinds of blood vessels we are talking about? We all belong to a system where we have a closed system, that is we all belong to a closed circulatory system. So we have blood vessels to carry the blood to different places. Now what are the different kinds of blood vessels we are going to look at? The first one, you all know this is an artery. The second one here on the screens is a vein. And then the third one is a capillary. Now, what is the difference between these three kind of blood vessels? Artery always carries oxygenated blood and we call it as a pure blood. But there is one artery which carries impure blood, that is the pulmonary artery. We will try to understand why this carries impure blood in the next session we are going to take up. So right now, pulmonary artery is the only artery which carries deoxygenated blood. The same way, here you have a vein, and this vein, except the pulmonary vein, all the other veins carry the deoxygenated blood, that is the impure blood. Now what is with the capillaries? Capillaries are generally very thin and they are found at the junction of arteries and veins. And how they occur, we will have a look at it. Now coming to the differences between arteries, veins and capillaries. On the screens you can see artery is generally thick and lumen, what you call the space inside, is very, very small compared to that of a vein. And vein, you can see it has a thin muscle and it has a lot of elastic fibers, but it has a very large lumen. Now this lumen part is very, very important because in arteries the flow of blood has high pressure. So it flows with a lot of pressure, as well as it is intermittent flow. That means it is not a continuous flow when it comes to arteries. Now, so it just leaves, it gives gaps for flowing in that particular place. Whereas in vein, it is a continuous process, the blood flows smoothly. Now this will be the difference, major difference between these two. This happens because of the lumen, which is smaller in arteries and larger in veins. Veins do not have that much pressure to continue the flow. So valves are generally present in the vein region, whereas in arteries, you do not need any kind of valves or support because they already are flowing in that particular direction. Now capillaries, let's look at the capillaries. The capillaries are all one-celled thick. You're already given here. It's a single layer of cells and it's extremely thin. They are very, very thin because capillaries are the areas where you find that there is exchange of any kind of substances or any kind of gases. Whatever the exchanges happen, it happens only through the capillaries, because the capillaries will be the portion between the arteries and the veins. Now, how do these arteries and veins divide? the arteries will continue to divide into the arterioles. Now these arterioles will join with the capillaries. Now these capillaries, all of them will join together to form a venule, and all the venules together become the veins. Now you can see how it does. Now arteries always break down into smaller structures which are called as arterioles. Now these arterioles break down into capillaries, and once the oxygen or the substances have exchanged at the capillary level, from the tissues, the capillaries will pick up the carbon dioxide or any kind of substances which has to be given out. Now all that will be carried through the venules, and venules all will join the veins. So the junction of arteries and veins are the capillaries. Now they are a very, very important part of the system because that is where the exchange takes place. So when we did the breathing in gases session, we talked about capillaries being there in the alveoli region, where there is exchange of those kind of gases between the tissues and the alveoli. So there also we talked about lots of capillaries to increase the surface area at that junction. Now this is all about the blood vessels. To summarize this, just look at the differences here. Artery always has a thicker wall, whereas a vein has a thinner wall, and capillaries are generally one-celled thick. They have a smaller lumen, and veins have a larger lumen, and capillaries very small lumen because the size itself is very, very small. Now arteries, blood always flows with greater pressure or high pressure, and vein has less pressure in its flow, and capillaries are all single celled. It's just for the exchange of that particular place. You can all have the thicker regions, and arteries generally carry pure blood except the pulmonary artery, whereas pulmonary veins generally have the deoxygenated blood in them except the pulmonary vein. So arteries always carry the blood away from the heart because heart is, after purification, it is carried to various parts of the body. But veins always carry blood towards the heart with the exceptions we have already taken out. Now this is all about the blood vessels and the differences. And coming to the next one. So we understood what are the blood vessels and how this clotting factors occur. Now let's take something very interesting. Have you ever imagined what are your blood groups? You must have gone for testing during some procedures, and then you must have known that you have certain kinds of blood groups. Now blood groups, all of them form a chain of groups which is called as the ABO blood group. Now this ABO blood group, why do we give this? Because we have A blood group, then we have the B blood group, and then AB and O. Now why did we say O here? O is a factor because O doesn't have any of the A, B or AB. Now what is A, AB and A? So on the screens you have seen here, it is written something as antigen and antibodies. Now antigens are the substances which are generally found on the surface of RBC. Now with this RBC surface, whatever antigen, whatever is found on the surface, the antigen, that gives the name of the blood group. So for A blood group, you have an A antigen, and for a B blood group, you have as the B antigen, you can see it here. And then for AB, you have the AB antigens. Now this will define the blood groups. Now what is with the O? O doesn't have any antigens on the plasma region, on the RBC region. That's why we have given here, there are no antigens. Okay. Now coming to what are these antibodies? So whenever you know a microorganism attacks, we need to produce as a defense mechanism antibodies. Similarly, blood also needs that antibodies to protect the antigens. So these antibodies are always opposite in the coding. That is, if you see an A antigen or A blood group, the antibodies on the plasma. So antibodies all come from the plasma region. Now this antibody production for A will be B, and for B it will be A. Opposite we see antibody A, antibody B, we have given here. So A blood group, B antigen, B antibody, B antigen, you have A antibody. But since AB blood group here on the screen, so AB blood group has both the antigens, they cannot produce any antibodies as such because already they are having both the type of antigens. So that's why there are no antibodies present in the plasma in AB blood group. Now what about O? O, since it does not have any antigens, it is free to have all the antibodies in its region, that is in the plasma. So the O blood group will have an anti-A and anti-B, that is antibodies A and B, both are present. Now this is how we actually name that particular blood groups. Now then you will see that it is just not AB and O. Sometimes it is given as positive and negative. So this is also one of the important things because we see A positive, B positive, AB positive, O positive like that, or negative blood groups. Now what is this positive and negative we are talking about? Positive is a rhesus factor. Now this factor comes from the monkey of the same name, that is the rhesus, where it has a D antigen. So it has one more antigen called the D. Now if there is a D antigen along with the antigens you are seeing, then the blood gets positive. If there is no D antigen, then we give it as negative. So that will be the definition of having this particular positive and negative. Negative blood groups are very rare because 1 in 10 may have that negative blood groups. Positive blood groups are more common in the population because negative is very, very rare. You cannot have that blood group in a regular fashion. Now coming to that understanding, let's look at what are the universal donors and universal recipients. So what is meant by a donor and what is meant by recipient? Donor is some blood group which can always give blood to any kind of blood group. Now universal donor will be the type O because it has no antigens at all. It is free to give blood and you can receive blood from O, any kind of blood group. So type O, we call it as a universal donor because it can donate blood to any kind of patient. And type AB, because it has no antibodies as such, we give the term universal recipient because it can accept blood from any groups. So type O, universal donor, and type B, we call it as universal recipient. So this will be the understanding of the blood groups. Now you can always ask the question, is it like only A blood group will be the one? So you can take blood only from this A or O? Yeah, because if I am looking at a blood group A, then I can receive blood either from my own group or from an O because it's a universal donor. Now the B can receive from B and O, but AB can receive blood from A, B and AB because itself and the other two because it has both A and B antigens in it. So now this blood group we can accept from any other. That's why we gave the term universal recipient. Now what is with the O blood group? O blood group can donate to every other blood group, but it can take only its kind. So it is only a donor and you cannot accept blood from any other blood group. So O can accept only its own kind. Now then comes the positive and the negative. So positive blood. Now remember the positive blood people, they can take blood either from the negative or the positive. But what happens is when you have a negative group, there might be mixing if you are donating blood to somebody. So that what happens, then there is a problem because there is a plus and a minus involved, which is very, very problematic, especially when the mothers are having such kind of babies in their wombs. So the negative blood cannot mix always with the positive blood because it produces that kind of effects. So positive blood is much easy to have these options than the negative blood. Now this is all about the blood groups. So trying to understand this, there are 4 different types of blood groups, A, B, AB and O. So A blood group, it all named because of the antigens they are having, and antigens, the opposite of antigens will be the antibodies, and antibodies will be present on the plasma where the antigens are present on the surface of RBCs. Now since whatever antigen is present, the name is given as such to the blood group. So coming to this, and universal donor is O blood group and universal recipient is AB blood group. Now, so that's all with this blood groups here. So this one will be a part of the system what we have taken just now. Now let's look at some of the questions So we will look at some of the questions which can be answered. Okay. Now there is a question here, why are there valves in veins and not in arteries? So this we already studied here. We said arteries have this high pressure in that particular flow. So the veins do not need they need valves because the pressure doesn't maintain their continuity. Since arteries flow with blood pressure, the blood flows with blood pressure, you don't need valves in the arteries, but you definitely need, the veins definitely need valves. Because you cannot have backflow of blood, and you all know veins are all made up of impure blood. So they cannot go and join that artery, so that mixing of blood happens at that particular place. So veins always come with valves. It is toxic also. It is toxic also. What factor is needed for the conversion of fibrinogen and fibrin? We already saw this. We said we need certain substances like the calcium and vitamin K, which will help in the initiating the process of that particular one. But for the fibrinogen to be converted, you always need that thrombokinase enzyme also. Now the enzymatic reactions which all help you to convert the fibrinogen into fibrin, which is the clot what we are talking about. Now what are the capillaries and what is the role they play in circulation? Capillaries are the areas where the exchange of substances or materials occur, whether it is digested food or the gases. So that is in between the arteries and the veins. The arteries join the capillaries, and then the capillaries join together to form the veins, and that's how the entire circulation happens. So capillaries help in exchange of all these materials. Now coming to few more questions. What is hemophilia? Hemophilia is otherwise called as the bleeder's disease. We have taken this. Now bleeder's disease happen because there are no clotting factors and the blood does not clot. So that is what happens in hemophilia, and this is a special kind of genetic disease also. It runs in the family. Now coming to the next one. Trace the pattern how arteries and veins circulate in the body. We have given the flow chart already. So the arteries break down into arterioles and arterioles join to form the capillaries or break down into capillaries. Now these capillaries join together to form the venule, and all the venules join together to form the veins. Now this will be the circle, and capillaries will be in between this kind of junction. Okay. Now this is all about the tracing the arteries. You can draw it, or either you can give the flowchart diagram and you can explain how they break down. Now coming to the last question. Name the different blood groups found in humans. Why the blood groups contain a positive and a negative sign? So looking at the blood groups, so just name them. What are the different blood groups? You can give the names here, A, B and O. And why they have a positive and a negative? This comes from a factor which is called as a rhesus factor. And it contains another antigen what is called as the D antigen. Now this D antigen will, if it is there, it is positive. If it is not there, then it describes it as negative. So it comes from the rhesus monkey, which is also called as the rhesus factor. It's given here. Now this is all about the questions to be answered. And for now, this will to just or give a summary of what we studied today. We looked at the different blood groups, then we looked at different kinds of blood vessels which are a part of the circulatory system. And we also understood how clotting occurs and why clotting is very, very important for stopping that flow of blood. Now coming to the next session we are going to take up. Next session we will be looking at the human circulatory system. We will be studying the parts of the heart and its other, how the heart works, the functioning of all that. And that will be for the next session. So keep watching, stay tuned with us, and then thank you so much for watching this session.